In Chapter 4, we will discuss the ways in which living organisms get the energy necessary to maintain life. Energy is needed for every action in the body, so it is important that organisms are able to harvest what they need to function. The ultimate source of energy for all organisms on Earth is the sun's radiant energy. This energy is directly processed by plants or producers, which are then eaten by animals or consumers. Eventually, decomposers break down any remaining particles, which are released as nutrients and heat. Metabolism is the total of all the chemical reactions which happens inside cells that either consume or generate energy. When talking about metabolism, there are two types of pathways: anabolism and catabolism. Anabolism is taking smaller, simpler molecules and making larger, more complex molecules. These reactions require an input of energy to build bigger molecules. Some of you may have heard of anabolic steroids. If you think about that for a minute, why do some athletes want to use them? It is to build up their muscles into something bigger. So although I do not condone their use, I hope this helps you remember what the term anabolic means. Catabolism, on the other hand, is the opposite. It takes big molecules and breaks them into smaller, simpler molecules. And just as in the opposite of anabolic reactions, catabolic reactions release energy. To help you remember that catabolic reactions break down molecules, consider the prefix cata, which means, among other things, down or downward. Think about the word catastrophe. What does it usually involve? One example that is easy to understand could be a catastrophic storm that destroys a neighborhood or a city. Hopefully this connection will help you keep these two ideas clear. The study of energy and energy transfer is known as thermodynamics. Energy can be transferred in either a closed or an open system. The diagram on the left illustrates a closed system where the object does not gain or lose energy to the environment. With living organisms, they are constantly exchanging energy either as a gain or a loss with the surroundings. The first of two laws of thermodynamics which we will discuss is that energy cannot be created or destroyed. All energy that exists now has always existed and will always be conserved. However, energy can be converted from one type to another to make it usable for an organism or for a cell. In the diagram, the energy stored in the molecules of ice cream is converted into energy for kids to move and play. Plants can capture light energy from the sun and turn it into the chemical molecules of glucose or sugar, and therefore create necessary energy to survive. Although the first law of thermodynamics makes energy usage and transfer seem simple, it is more complicated because the system is not fully efficient. Every time energy is transferred, some energy is lost that is not usable, and that is typically lost as heat energy. So the second law of thermodynamics states that some of the energy will be lost to the surroundings. A measure of randomness and disorder is known as entropy. A living organism that is highly organized and maintains energy input and output is considered to have low entropy. A system that loses more energy to its surroundings is considered to be less organized and therefore have high entropy. Notice in the diagram the circles that are well organized on the left would be considered to have low entropy, while the disorganized random circles on the right would be considered to have high entropy. Potential energy is the energy available as a result of a position or gravity, while kinetic energy is the energy of motion or work being performed. As energy is a transformation, it transfers from full potential in the stretched out bow position to kinetic energy when the bow is released and pushes the arrow forward. To better illustrate the transfer of energy, look at the pendulum. At the far left, at position 1, the potential energy, or PE, is at its maximum as it is as high as it can go, and therefore the most possible energy is stored. At that same location, the kinetic energy, or KE, is 0 because the pendulum is not moving at that location. As the pendulum swings, it loses potential and gains kinetic. At position 2, they are both at 50%. Then at the bottom, where it would be moving its fastest, kinetic energy is at the maximum and potential is used up at 0. It will lose kinetic as it goes back up through 4 and 5, but gain potential as it gets to 0.5. Then as it drops, it goes back through the same transfer of energy. Any molecule that has bonds has stored energy in those bonds. When those bonds break, energy is released. Any food that you eat is full of chemical bonds. When those bonds are broken, you are supplied with chemical energy, which is food for your cells. After a chemical reaction occurs, some energy is lost as unusable and some energy is not used but is still able to do work in the cell. This energy is known as free energy. When a reaction releases energy into the environment, it is known as exergonic reaction. Energy is exiting the reaction. Even when a reaction releases energy, however, it still will require a bit of startup energy known as activation energy. However, when energy is required for the reaction and therefore is an input, it is known as an endergonic reaction. In this case, energy is entering the reaction. In an exergonic reaction, there's a net loss of free energy. So these curves are shown as a downhill curve. The products or end results have less free energy than was present in the reactants or the beginning substances. These reactions can proceed spontaneously. In an endergonic reaction, there is a net gain of free energy and the curve is shown as uphill. In these reactions, the products or end results have more free energy than was present in the reactants or the beginning substances. A certain amount of activation energy is required to initiate every reaction, even a spontaneous one. So, even exergonic reactions that will be releasing energy require a little input of energy to get things going. This energy is known as activation energy. Notice in the diagram on the left, the overall curve of the graph is downhill, but before the reaction was complete, the curve goes uphill as an indication that energy was required to complete the reaction. The blue arrow shows the amount of energy needed to make the reaction take place. This rise in energy is the activation energy, as it is the energy that activated the reaction. In the diagram on the right, which is an endergonic reaction, the energy of products or end results are higher than the reactants. The reaction is gaining energy, but in order to make the reaction happen, it needed a certain amount to activate the reaction. As a comparison, look at the difference in the two diagrams comparing the products to the reactants. Notice you can tell in an exergonic reaction on the left compared to an endergonic reaction on the right based on the ending point of the products. Many chemical reactions require activation energy that take a very long time to complete. Molecules that can help lower the activation energy and therefore speed up a reaction are called enzymes. Without enzymes, life would cease to exist. Enzymes are unique in that they are not consumed by reactions like most molecules and can speed up multiple reactions as a result. Notice in the graph, the purple curve is the natural activation energy needed for the reaction, but with the use of an enzyme, this amount is lowered. Notice the red curve, but the reaction can still occur. Looking at this curve, would you call it an exergonic release of energy or an endergonic gain of energy reaction? If you said endergonic, you were correct. The products have a higher energy level and therefore the reaction is endergonic. For an enzyme to speed up a chemical reaction, it must first bind to a reactant or a starting substance known as a substrate. The binding location of the enzyme and substrate is known as the active site, and this is where the action of the chemical reaction occurs. When the enzyme and substrate break free from each other, a product is released, and the enzyme is available to bind with other substrates, continuing to make more product. Although the enzyme substrate binding action works to speed up the reaction, the enzyme and the substrate do not always have a perfect fit. When they bind, the shape of the enzyme can change, resulting in chemical bonds which are more easily broken, which is ideal for the enzyme to perform its function of more quickly producing a product or products. This change in shape to a form fit is considered an induced fit. By the end of the reaction, the enzyme returns to its original shape and can continue to participate in speeding up reactions. Activity which can either promote or reduce enzyme activity is called inhibition. In the case of competitive inhibition, the inhibitor competes with the normal substrate for the active site of the enzyme. A competitive inhibitor occupies the active site only temporarily, as in the top diagram, enzyme-substrate complex labeled A. In the bottom complex labeled B, the inhibitor binds with the enzyme at a site other than the active site, but still altering the shape of the enzyme and therefore inactivating it. This is known as non-competitive inhibition because it does not use the active site for binding. An allosteric site is a location other than the active site where an inhibitor can bind that changes the shape of the enzyme, thereby preventing the substrate from binding, which is occurring on the left side of this diagram. The opposite can happen too, as in on the right side of the diagram, when an activator instead of an inhibitor binds to an allosteric site, altering the shape, allowing a substrate to bind. This time it activated the active site instead of blocking the active site through reshaping. Some enzymes need a helper molecule to obtain the optimum shape for the substrate to bind. These molecules are cofactors and coenzymes, and just like enzymes, can retain their shape and be recycled many times. Cofactors are inorganic ions, which if you'll remember are charged atoms, and coenzymes are organic molecules made of hydrogen and carbon atoms. One more type of inhibition used by cells is known as feedback inhibition, where the enzyme's activity inhibits the end product. Cells will use this type of inhibition when there is plenty of end product and no more is necessary. Notice in the top diagram, the green triangle is the substrate or beginning reactant for enzyme 1, which then produces a purple rectangular product. That purple product becomes the substrate for enzyme 2 and down the line until the end product of an orangish yellow square is produced. If the environment gets enough of the square, the pathway can be inhibited by its own end product. Now, notice in the bottom diagram that the yellow orangish square will attach to an allosteric site, a site other than the active site, near the beginning of the pathway and can change the shape of enzyme 1. When this change happens, the substrate is not able to bind to the active site, thereby inhibiting further product down the line. Energy production in the cell works in this manner. If there is too little, the pathway breaks down glucose to form ATP, but once enough ATP is in the pathway, then the same ATP will attach to an allosteric site and shut down the process of glucose metabolism, and therefore the production of ATP is stopped. Watch the linked video for an animation of this process. Enzymes are highly beneficial to cells performing work. However, they require an ideal environment to best do their job. If the temperature of the environment changes, it impacts the enzyme's ability to function by changing its shape. The same is true with pH levels and salt content outside of the enzyme's preferred range. When these levels change, they too can alter the shape and therefore the ability of the enzyme to function properly. When the shape change is irreversible, it is known as denaturation or being denatured. ATP, or adenosine triphosphate, is the energy currency in cells. It provides energy when one of the phosphate group bonds is broken. To better understand this molecule, it is important to understand other similar molecules. AMP, or adenosine monophosphate, is similar to ATP, but only has one phosphate group, and ADP, or adenosine diphosphate, has two phosphate groups. When AMP and ADP gain phosphate groups, there's a high level of energy stored in the bonds. So when ADP or ATP lose a phosphate group, energy is released as a result. It is ATP that is the goal of cellular respiration, also known as aerobic respiration if oxygen is present, or anaerobic respiration in the absence of oxygen. To understand the process of cellular respiration, it is imperative to understand the molecule NAD, which is an electron carrier or coenzyme that helps in the chemical reaction. NAD is an abbreviation for nicotinamide adenine dinucleotide, and depending on its charge can be positive or neutral, which changes how it is written, either with a plus sign or with an H on the end. The charge difference happens due to an oxidation reduction or redox reaction. This change in the molecule's charge is due to two hydrogen atoms, two positive protons and two negative electrons, being gained and/or lost during the reaction. One way to remember redox reaction gains and losses is the abbreviation OIL-RIG, which means oxidation is losing electrons and reduction is gaining electrons. As electrons carry energy, oxidation is a loss of energy, while reduction is a gain in energy. Notice in the diagram just below the ribo for ribose, there is an N positive on the left and just an N on the right. Now look at the equation in the middle of the two molecules. NAD positive plus H positive plus 2 electrons. This is gaining 2 electrons and is then written NADH. Both NAD positive and NADH are actually the same molecule, just with a different charge. NAD positive is a low energy molecule, and NADH, because it has gained electrons, is a high energy molecule which helps drive cellular respiration. This same concept is true with the electron carrier flavin adenine dinucleotide, or FAD. You will see FAD in the third step of aerobic respiration, the citric cycle. Watch the two linked videos for a visual explanation of redox reactions of NAD. The stages of aerobic respiration occur in specific locations in the cell. Glycolysis, the first stage, occurs in the cytosol, the gel-like fluid of the cytoplasm of the cell. Pyruvate, the product of glycolysis, enters a mitochondrion where cellular respiration continues with the formation of acetyl-CoA, the citric acid cycle, and then electron transport and chemiosmosis. Most ATP is synthesized by chemiosmosis. The first phase of aerobic respiration happens in the cytosol of the cell. It is the only step that does not happen in the mitochondrion. The first phase of this process starts with a 6-carbon sugar molecule of glucose and is catabolized or broken down, and in the second phase of glycolysis produces 2 3-carbon sugar molecules known as pyruvate or pyruvic acid. The first process uses 2 ATP, which is endergonic, while the second produces 4 ATP, which is exergonic, for a net yield of 2 ATP. 4 minus 2 equals 2. It also produces 2 NAD molecules, which will be needed in a future step. Although glycolysis is a step in the aerobic respiration process, this step does not require any oxygen. Watch the video for an animated version of this process. Now that glucose has been broken down from 1 6-carbon molecule to 2 3-carbon pyruvate molecules, the next step in aerobic respiration is the formation of acetyl coenzyme A. This is the first step that happens inside the mitochondrion. In the mitochondrial matrix, a carboxyl group highlighted in blue is removed from pyruvate and released as a molecule of carbon dioxide. This CO2 is a waste product and will be released from the organism. What is left is a 2-carbon molecule. In step 2, the 2-carbon molecule from step 1 is oxidized, meaning it loses electrons, and the electrons lost in the oxidation are picked up by NAD+ to form NADH, which is high energy and will help drive the next steps of the respiration process. Finally, in step 3, the oxidized 2-carbon molecule, which is called an acetyl group and is highlighted in green, is attached to a helper molecule called coenzyme A. Together, the acetyl group and the coenzyme form a molecule known as acetyl-CoA, which will help carry the acetyl group, most importantly the 2 carbon atoms, into the citric acid cycle. Each original glucose molecule is catabolized or broken down into 2 pyruvate molecules. Each pyruvate produces 1 acetyl-CoA, so this process results in a total of 2 acetyl-CoA molecules. Now that glucose has been catabolized or broken down into acetyl-CoA, the carbon molecules can enter the citric acid cycle. This cycle is also known as the Krebs cycle and occurs in the mitochondrial matrix as well. This step requires oxygen, therefore if no oxygen is available, the cycle does not complete. Also notice that the citric acid cycle is a closed loop, meaning that it produces the molecules needed within the cycle to continue. The citric acid cycle is an 8-step process that starts with one of the 2 acetyl groups that was delivered by the acetyl-CoA from the previous step. For every glucose molecule we started with, 2 acetyl groups enter the citric acid cycle, as seen at the top on the left diagram. Each 2-carbon acetyl group combines with the 4-carbon compound oxaloacylate to form the 6-carbon compound citrate. During each turn, 2 carbon dioxide molecules are removed and energy is captured as 1 ATP, 3 NADs, and 1 FADs per acetyl group, or a total of 2 ATPs, 6 NADs, and 2 FADs per glucose molecule. Remember that these molecules are high energy and will help drive the rest of the aerobic respiration process, which will ultimately produce a large amount of ATP for the cell to perform necessary functions. The diagram on the left illustrates each of the 8 steps in the process of converting an acetyl 2-carbon molecule into the various energy and waste molecules. On the right is a simplified view of the conversion of pyruvate or pyruvic acid molecules through the citric acid cycle. It shows the input and output of each phase. Watch the video for an animation of the citric acid cycle. And finally, we are to the final step of aerobic respiration, which produces the most energy. we are going to look at how the electron transport chain works and what chemiosmosis is. This step of aerobic respiration occurs in the mitochondrial membrane. There are enzyme complexes which carry electrons across the membrane and pump hydrogen ions into the intermembrane space. The blue arrows indicate the pathway of electrons, while the white arrows indicate the pathway of protons. This creates something called a chemical gradient, where more hydrogen ions are on one side of the membrane than on the other side. This uneven distribution of ions then results in diffusion of those ions back across the membrane into the matrix through the last enzyme complex known as ATP synthase. This diffusion of ions across a membrane is known as chemiosmosis and provides the energy for ATP synthase to rotate. When the central structure of ATP synthase rotates, ADP is combined with inorganic phosphorus, represented by the capital P lowercase I in the orange circle, to then form ATP. 2 hydrogen ions and a half of an oxygen molecule will form 1 molecule of water. This oxygen atom serves as the final electron acceptor in the process and is the reason it is referred to as aerobic. The entire aerobic respiration process can produce 32 to 34 molecules of ATP, the ultimate source of energy for cells, depending on the organism and the tissue in the organism. Watch this video to see an animation of the electron transport chain. The overall yield of ATP as a result of aerobic respiration is shown in this flow chart. Notice most ATP is produced by the electron transport chain and chemiosmosis in this last step. The opposite of aerobic respiration is anaerobic respiration, which is the production of energy in the absence of oxygen, such as which occurs with fermentation. In the process of fermentation, a molecule of glucose will be broken down into pyruvate through glycolysis, just like in aerobic respiration. However, if oxygen is not present, the result is either lactic acid, as which happens in animals, or ethanol alcohol, as which happens in yeast. This process is very inefficient as it only produces 2 ATP from the glycolysis phase in comparison to 34 or so from aerobic conditions. Watch this video for an animation of fermentation. Glucose is not the only input source for cellular respiration to produce energy in the form of ATP. Glycerol, which is a long chain of glucose, can be stored in the liver and utilized when sugar is low in the bloodstream. Proteins, which break down into amino acids, as well as lipids such as cholesterol, which break down into fatty acids, can all enter the cellular respiration pathway and be catabolized into ATP molecules, providing energy for the cell when glucose is low or absent.